it's a pleasure to be at this place, which is putting together kind of finishing touches on this project that started quite a while ago, but it almost feels like it was just yesterday. So of course, this project was the first one funded under the New Jersey RMI program. We were really proud to get out of the gates early. Some of you, if you were on the steering committee at the time, might remember there was some urgency in getting started because of steel prices of all things. And because we were building the dredge and trying to get some things done in time for a side-by-side survey with the feds. We were very happy to be able to get things started when we did. And I think we've had a lot of success because of the way that we kicked things off. So this project, as Caitlin mentioned just now, the thrust of this project was setting up capacity to do Surfclam surveys with a really specific piece of science equipment, fishing equipment, and then also leveraging some of the time at sea to do some really novel and exciting carbonate chemistry experiments with Surfclam habitat and biological responses. The team on this one, me, Daphne, Jason Morrison at the time was at the Haskin Shellfish Research Lab. He's now at NOAA in the Cooperative Fisheries Division, but has stayed on as project kind of team partner, even though he's moved over to NOAA. Sarah Borsetti, who's on the call, of course, Dr. Grace Saba up in the Department of Marine and Coastal Sciences is a project partner on this. She is an expert in ocean acidification. And so helped us with that side of the project. Captain Tom Dameron from Surfside has been an industry collaborator on several things and really, really instrumental in this project. And of course, Dr. Dan Hennon, who is this stock assessment survey lead for surf clams and ocean quahogs at the Northeast Fisheries Science Center. And his partnership in this was really critical to make sure that we were really setting up our surveys and our equipment and in a way that would walk hand in hand with the way that federal survey data is collected. And of course, Caitlin, who's on the call, and Colleen Brust from the New Jersey DEP have been amazing in helping us get all of this work done and contracted and giving us advice along the way. So the way I'll describe things, the goals of the project, of course, was to build a scientific dredge. This dredge has kind of some unique characteristics I'll talk about in a second that make it a really great science tool for us. And then to calibrate that piece of equipment. These calibration experiments are big and costly and sometimes a little risky. And I was really happy to have success in all of the calibrations that we accomplished. And then our third objective had to do with some of these ocean acidification questions. And so I'm going to start off talking about, first of all, some of the survey supportive tools here. The first of which was building the dredge. This picture on the right, you can see our science dredge on the back of the Joey D. Compared to the dredge on the left. That's a regular fishing dredge on the left. It's maybe a little bit hard to see in these photos, but the bar spacing between the metal bars of the box of the dredge are narrower in our dredge, and that allows us to catch smaller clams, which of course the fishery doesn't want to do because that's below the legal size limit, but we want to see those animals because they're part of the population that we're trying to assess. The advantage of doing this in this way, rather than lining the dredge with chicken wire or some other kind of smaller mesh liner, is that the dredge then still fishes efficiently like a fishing dredge would and doesn't catch a lot of the stuff that we don't want to have to handle and see. And I will point out that the plans for this dredge were developed and designed by the fishing industry, by collaborators in the fishing industry. And those engineering designs were not part of this project. They were already in place before this project was funded by RMI. And those were funded directly by industry through the NSF Science Center for Marine Fisheries. So kind of a really interesting tie-in to industry relevance here. So this was a tool designed by fishermen who know how these things work. And I love this picture. This is always one that I use to say it worked. This is a picture of some of the different size classes of clams that this dredge catches. The smallest clams there on the tray would not be retained in a regular commercial dredge. So we are retaining those in that dredge. And I will say also that we were really happy to see that the catches came in relatively clean, so that our processing and handling times on deck weren't at the scale that you get when you have to line a dredge with chicken wire. So the first step in our calibration experiments was a side-by-side survey with the federal survey platform, the Pursuit, which is the boat that they use for the federal surveys, which is a big double dredge boat. One of the urgencies of getting this off the ground with RMI when we did was so that we could get the dredge built in time to get in the water and do this side-by-side experiment. And I will say we were successful. Dorchester Shipyard built our dredge, and they kind of expedited some of the construction to make sure that and I think they finished construction just about a month before we were out on this particular survey. You see the stations we occupied there with the triangles. All of those stations were part of the federal survey, and we were out there at the exact same time as the federal survey, which helps in data comparison. The track there is the track that we followed as we went and occupied these stations. And I'll point out that we specifically selected stations that overlapped with offshore wind lease areas. You can see the lease areas there in the shaded blue. We did that so that we could be collecting additional baseline data for offshore wind lease areas as a kind of a piggyback on this project. And this is a look at our survey operations on the deck of the boat. The picture on the left is looking towards the back of the boat. That's Ailey Sheehan, my lab manager. We set up a station back there. You can see behind her is our CTD. We were doing profiles of each of the stations before we took a dredge tow. So we were collecting oceanographic data, which is in addition to something that the federal survey does. They don't do these kinds of profiles. And then looking at the picture on the right is looking towards the wheelhouse, the front of the boat, and you can see the three people standing there are at our clam processing table. So we have a length measuring board and a shucking station there where we were processing the catch. This is a look at over on the other side of the boat. We were also taking benthic grabs. These were being done before the dredge tow was taken. These grabs were meant to provide us with information about young of the year or juvenile surf clams. This is, again, something that the federal survey doesn't do. And you see a picture on the left there, those two top clams, not the really tan colored one, but or mahogany colored one, but the other two are surf clams. This is something in addition that the federal survey doesn't do, but we were able to do with some success. This also is one of the pieces of data that the state, New Jersey DEP surveys for surf clams used to do when those surveys were going annually. So a couple of different data integration pieces here with the grabs we were taking. And so here's a look at some of the data for adult and young of the year surf clams. Each of those dots is colored by the number of clams caught at those stations. And again, so the point here is we caught clams at almost all. The open circles means that we didn't get clams. We caught clams at almost all the stations we occupied. We also got baby surf clams in most of the benthic grabs we deployed. And in some cases, we were getting very high abundances of adults or even juvenile surf clams at these stations. So a nice spread in some of the biological information we were getting. The point of this survey that went out in 2022 was to do paired tows with the federal platform, which was the Pursuit, shown in blue here. And these are just some comparisons of those paired tows in terms of biomass on the left, surf clam biomass this is, and surf clam abundance or numbers per meter squared on the right. And so these are standardized. In both cases, both survey platforms perform tows in the same way. So we were using 5-minute tows. But then also standardizing those tows to the actual bottom that was occupied by the dredge so that we could calculate a clams per meter squared number out of both of those surveys. The point here is we were getting pretty comparable numbers. In some cases, the Joey D caught a few more clams than the Pursuit. In other cases, vice versa. But in general, we were getting very, very similar estimates of abundance and biomass from the two platforms. The one nuanced difference between the Pursuit, the federal dredge, and our Joey D smaller bar spacing dredge is the size distribution of clams that were caught. So what you see here in this histogram is, and the probability distribution functions in blue is the size distribution of the clams being caught by the Pursuit, and then in orange are the sizes of clams that we were catching with the specialized dredge. We expected to be catching clams that were smaller because of the narrower bar spacing. And in fact, you do see some of that in this data distribution. What we didn't expect is that the Joey D was also catching clams of a larger size than the Pursuit. And Dan, Sarah, and I have all had long conversations about what might be going on here. But the bottom line is we know that size distributions of animals out in the real world aren't a bell curve or a normal distribution like you see for the Pursuit. Instead, we expect to see kind of cohorts showing up in the distribution. And in fact, that's what the Joey D reflects. So we think that the Joey D is doing a better job of capturing a representation of the true population that we're observing in terms of size frequency. So we were really happy to see that. One of the other things we were doing with these samples is we were looking at ages. We brought back a subsample from every dredge tow in the 2022 survey, and we aged those clams. What we do to age them is we section them through the umbone, which is the hinge side of the shell. We section through there with a diamond blade saw. And then we use some fancy polishing tools to polish this chondrophore, which is just that little process inside the hinge. And when you polish it up and then look at it under a microscope, you can see very clearly annual growth lines in the hinge. And so for each of these animals, we then knew the length of the shell. We knew how big the animal was at the time it was caught, and we also know how old it was at the time it was caught. And then we can put all of those numbers together, and we can come up with what's called an age at length, or a length at age, or a function that describes growth on average across this population. And I will say that, and I'll talk about this at the end of the presentation, but this data set has been very fruitful, and we've been using it for a number of follow-on kind of student research activities and things to look at differences among growth rates at depth, or differences among growth rates in different regions or densities. And so this was a really powerful data set that we were able to collect in the 2022 survey. And I will say that also the federal survey didn't age all of the animals from all of the stations they collected animals at. So this was above and beyond what would typically be done. One of the fancy things, now moving on to some of the 2023 experiments to further kind of explore how this dredge was collecting clams. One of the fancy things that's built in the design of the dredge is the bar spacing not only is narrower on the bottom of the box. This is looking into the box. It's at the shipyard in winter storage, but it's looking at the bottom of the box. These bars can actually be ratcheted apart so that they can be opened up to a spacing that is as wide as a commercial dredge. So basically it's got variable bar spacing. In all of our survey tows, we were using the bars in their narrowest configuration, but we can in fact open it up to a wider bar spacing. And so we did take samples in 2023 with at 10 stations where we had the bars in the narrow configuration, and then those same 10 stations where we had the bars opened up in open configuration. And that allowed us to explore this size selectivity of the dredge a little further. These two size frequency histograms are those two configurations, and these are the lengths of clams that were collected across all 10 stations that we occupied. The purple bars are the closed down spacing. And so, of course, we do see smaller clams being caught when the bars are closed. And then the green distribution is when the bars are open. And interestingly, we catch even bigger. And if you remember, we're catching bigger clams with the bars closed compared to the Pursuit. But with the bars open, we yet again take another step into catching, more effectively catching big clams with that open spacing. So again, this dredge pretty effective at catching kind of what we think is a realistic distribution of clam sizes. Um, I'll move on now to the second experiment, the third experiment of the project, but the second experiment that we did in 2023. Again, we went out with the Joey D and we performed what are called depletion experiments. We did these at these 8 locations shown in the circles on the map. Again, we tried to leverage the fact that we were going out to do these experiments to collect additional data about clam populations in offshore wind lease areas. So you see that 5 of those experiments were located within the wind lease area footprints. In these experiments, what we do, these experiments are really all about the skill of the captains and the that are running these boats. In these experiments, what we do is we constrain. We put, you see Captain Eddie here on the left. He's looking at his plotter, and that pink strip that is on the map there for him, that's the area that we said we need you to tow the middle of this box. So have the dredge go down at the beginning and then tow to the end and haul back. On the right, you can see some of those tow tracks and the pink box that the captains were towing within. And we do that because these depletion experiments, the idea is you tow a track and then you try to overlap that track and you keep wanting to overlap and overlap until you've depleted all the clams. The idea is on the first pass, you never catch all the clams, but you want to know how many clams you missed. So you have to go back and you have to keep towing and towing until you deplete those tracks. And of course, you're in an ocean with a big boat, and so it's really hard to hit the exact same bottom over and over again. So we did some things to try to maximize that. The box that we allowed the captain to tow in was 50 meters wide by 500 meters long, which is very, very small compared to some of the other experiments that have been done in the past, which made it very, very hard on the captains. And they did great. And you can see this is one of those tow tracks all overlapped here in the red box. So they're towing quite nicely down the middle. And then as we're going through the experiment, you need to keep track of what's going on in order to know when the experiment's done. When have you depleted to a sufficient amount to be able to calculate the efficiency from the data you've collected? It's actually a harder thing to do than one would think. So we were tracking catch volume and catch count of every single tow. That's those 2 plots in the bottom. And our metrics for completion of those experiments was we needed the catch volume and catch count to drop to 5% of the highest observed catch. And we also made an additional constraint that the asymptote of the depletion fit to those data had to drop to that same level. And so we had several kind of checks and balances to make sure that the experiment collected the data needed. It seems like I'm talking a lot about this, but it's really one of the harder things to do. And a lot of times in the past, people have assumed an experiment collected sufficient data, ended the experiment, went back to the computers at the lab and realized that they just spent 2 days at sea and didn't collect enough data. So we really wanted to make sure that we were getting what we needed to get, but not having to spend additional hours and hours at sea unnecessarily. The other thing we did that was really unique is we used a very, very high precision GPS unit that was mounted on top of the wheelhouse to give us very, very, we were getting location information that was precise to half a meter. And that was important because we wanted to really precisely know where our tracks were. And we were using GIS to map those tracks in real time up in the wheelhouse so that we were also estimating our overlap. So we knew that we were getting track overlap with our 2.5 meter wide dredge being towed behind the boat. And so after all of that, these 8 experiments produced the results in those gray bars on the left. Those are the efficiencies we estimated out of each of those experiments. And so we take a global average across that, and our estimated dredge efficiency for this dredge is 0.65, which is actually really, really close to what the estimated dredge efficiency for the dredge used by the Pursuit is. And that dredge for the Pursuit, I think, is 0.67. That efficiency was estimated over the course of about a dozen experiments. So we're really close to about the level of efficiency confidence that they have for that federal tool. I'm just doing a quick check on time. I think I'm going to end up running out of time here. So what's exciting about in terms of a bang for the buck from RMI, this tool that was developed, was built, and calibrated with RMI funds has since been used to survey 3 different lease areas. Atlantic Shore South was surveyed last year. We did 2 years of surveys at Ocean Wind 1 before that project was canceled. And we went down south and in collaboration with VIMS did a survey at the Seavow lease area, the Dominion project off Virginia. And in all cases, we're using standard sampling protocols that are consistent with what we used for RMI. And so we're pairing it with oceanographic data. And in some cases with these projects, pairing it with other fisheries monitoring programs to get kind of a regional and ecosystem level view of what's going on out there. And so we're pretty excited that the efforts with RMI were able to support these 3 surveys. Of course, things with offshore wind are a little bit uncertain right now, but there are also several other developers who've expressed interest in using our platform for clam surveys. And so with that, I'll move on to the other parts of the project, which had to do more with ocean acidification, characterizing bottom habitats, and then trying to link those to biological responses. And I will say, again, this is a really exciting kind of bit of frontier science. There's very little of this work that's been done in the real world. Most of the ocean acidification biological response data that we have in the scientific world come from controlled laboratory experiments, which we know isn't realistic. And so this was an attempt to collect some real world data on carbonate chemistry at the bottom of the ocean where clams live and collecting those biological samples to go with them. So the upshot of what we did is we used a PCO2 sensor along with several other oceanographic sensors to measure the bottom water carbonate chemistry when we were just before we took these dredge tows at various locations in both 2022 and 2023. The label on these graphics is a little bit incorrect. And then we did a dredge tow at that same location where we then collected shells, brought those shells back to the lab, and then did some regular morphometrics and aging on them. And then in addition, we did shell strength testing. So we used this machine that you can see Bree using there. Basically, it puts pressure on the shell until it breaks, and it measures very, very precisely the breaking strength of the shells. This is just a quick look at our shell strength versus the weight of the shell. Obviously, bigger, heavier shells tend to break at a higher force. And so for all of our metrics, because we're comparing across various sizes of clams, we standardized that shell strength. That's what the variable on the y-axis is here. It's a standardized shell strength. So it's the breaking strength divided by the weight of the shell. What you're seeing in these 4 panels is various environmental parameters, temperature, pH, PCO2, partial pressure of CO2, and then the derived state of aragonite saturation state, which is a calculated variable. And each of these dots is the average breaking strength or standardized shell strength at a given station, and then standard error bars around that relative to the environmental parameters measured on the bottom. And so in some cases, we saw a pretty big spread of some of these variables. We had pH that ranged from kind of the mid 7s up to low 8s, which is actually a pretty big spread in pH. But again, this was on the bottom, so we would expect that. We had quite a wide range of temperatures that we observed and aragonite saturation states. I'll say that generally we tend to think of aragonite saturation states around 1.5 and lower to be problematic for certain shellfish larvae or other critters. In this case, we really didn't see standardized shell strength correlated with any of these variables in a significant way. And one of the things that we think might be contributing to this is we had different age classes represented among our different locations differently. That's a lot of differences. But what we do know is when you look at standardized shell strength, this graph on the right, versus the age of a clam, you actually have, under a standardized condition, some of the strongest shells pound for pound in young animals versus old animals. And so we are taking a closer look at that. We do have ages for all of these animals. If we're taking a closer look at that to try to pull out this as a covariate You can think of this kind of as like young people have stronger bones than older people who have more brittle bones. So that's one of the things we want to continue with and expand on. We also have been working with a faculty member in the Department of Marine and Coastal Sciences, Dr. Fiorella Prada, who does a lot of work on climatic responses of corals and other shell-forming critters, including clams. And she has ways of measuring additional shell characteristics that we don't have the way to measure, and those are porosity and density. And so we're really excited about the potential to work with FIO and see if we can get some additional shell characteristics to bring into the mix to try to understand this biological response to what was clearly a varying bottom carbonate chemistry environment across all of our stations. And so we want to work with her on getting some of those measurements. And then we want to do some additional modeling that will allow us to maybe explain some of the spread we see in the shell characteristics, and of course, using age as a covariate. One of the other things that we've been doing a lot of work on, I alluded to this earlier, is looking at the ages of animals, and in particular, digging a little more deeply into incremental age. So what you see here is one of those cross sections of the chondrophore of a clam. You see each of the annual increments. If we know that that animal was collected in 2022, we can back into what year each of those increments is associated with. And then we can take relative measurements of those increments. As an example, you can see one of these increments here is relatively small compared to the next increment, which is relatively big. And then we're going to try to see if we can associate some of these relative changes in annual growth increments with oceanographic conditions. So things like was it a strong or a weak upwelling year? Was it a long or a short cold pool season? Some of these kinds of bigger oceanographic variables that we think we may be able to see patterns in the shells. And some of our shells are up to 25 years old. So there's a 2 to 3 decade time history recorded in these shells that we're pretty excited to take a look at. And I'll mention that Sarah Borsetti has done some of this work in the past with gastropods. Pretty excited about a couple of the things that we are still working on. But with that, I'll close out because I know I'm getting up on 30 minutes here. But huge thanks to our fishing industry collaborators who, again, were the ones who designed the dredge we used, which is an incredible tool now for us to measure clams, sample and measure clams. The captain and crew of the Joey D have been absolutely incredible to work with over the past 3 years, and definitely couldn't have calibrated the dredge without their skill and expertise. I have had a whole host of technicians and graduate students and staff from Rutgers and VIMS who participated in these surveys and other efforts to get us to where we are. So thanks to all of them, and of course to the RMI program for supporting this work and being incredible partners in all of this moving forward, and providing incredible feedback too on reporting and outputs from this. So I'll close there and take questions.